Thank you, Michael. That was great talk. The next presenter is Dr. Yaoliang Tang. Is Professor of Medicine Cardiology from Medical College of Georgia. Title of his talk is MIR 322 351 cluster engages in complex feedback interaction between notch and HIF1 signaling that controls fate of cardiac progenitors.
Thanks, Prasanna, for a nice introduction, and thanks Jay, Joe, and JQ for invitation. This is a really honor to be here, present my work. So, as we know, a single episode of myocardial infarction may lose about 1 billion cardiomyocytes, but right now the strategy for cell transplantation is a maximum like 10 billion cells, so it is not enough for heart regeneration. And the medical therapy like the bypass or PTCA can only make the blocked vessel pattern, but cannot regenerate that cardiomyocyte. And heart transplantation is the only approach for repair the heart function, but due to the shortage of donor heart and heart transplantation is not available for most of the patient. And we isolated cardiac-derived mesenchymal cell by the biopsy or the mixed heart tissue, and we can see the layer of fibroblast come out, and then the round bright cell migrate out, and then we can use the SCAR1 to purify the cardiac mesenchymal cell, and we found that this cell population is CD105 positive and c-KIT and CD45 negative cell. And this cell is quite unique compared to the bone marrow-derived mesenchymal cell because it expresses GATA4 just as Tim mentioned. It is a unique tissue-specific fibroblast cell, and it expresses GATA4 cardiac early transferrin factor at a different level. Right now, the most challenge of cell transplantation is the environment, because when you inject the cell into infarct heart, it is ischemic environment and poor blood supply. And the most important is we do the cell transplantation and we digest the cell, and the cell lost the contact. And I think this is important for their survival. And notch is for cell-cell contact, and the donor cell has the 5 ligand, JAG1, and DLL134, and the recipient cell, they have 4 notch receptor, notch 1, 2, 3, 4. When they combine together, and the gamma secretase will cut the notch receptor, and the intracellular domain will enter into the nucleus, combined with transferrin factor RPBJ, and activate proliferation and survival signaling. And today I want to know how notch signaling modulate mesenchymal stem cell or fibroblast cell differentiation, and then next I want to see how notch signaling mediate the paracrine factor, and finally I want to see what is the downstream microRNA regulated by notch signaling. First, to know how notch signaling modulate mesenchymal cell differentiation, and we do the PCR array that cover all the tissue, and we find that the most upregulated is a smooth muscle, vascular smooth muscle gene, and endothelial gene. And we found that myosin heavy chain 11 was significantly upregulated at the mRNA and the protein level. And this upregulation is RPBJ dependent. When we knock out RPBJ, and we found that this notch-induced upregulation is disappeared. And next, we want to know notch-mediated mesenchymal stem cell paracrine factor. We collect the supernatant and do the protein angiogenetic factor array. We found that it can significant tons of angiogenetic factor like the angio1, DLL4, and ADM, TS1, PD, CGF. When we do cell transplantation, we found that compared to the media control, GFP control, and notch overexpress the cardiac mesenchymal cell, they can significantly improve the transplanted cell survival and also promote new vascular regeneration in the border zone of the infarct heart. And then next, we use the CT microspectra to check the perfusion, and we found that notch-modified cardiac mesenchymal cell, they can significantly improve the heart perfusion in the infarct area compared to the GFP control cardiac mesenchymal cell. Next, we used the micropeta, and we found that this notch modification the cell can improve survival the cardiomyocyte metabolism and reduce the defected area. So it means it can improve survival the hibernating the cardiomyocyte their function and make they have function after angiogenesis. Finally, we want to know what is the notch regulator microRNA. First, we want to know if notch regulator microRNA is important or not. So DISA is control the microRNA maturation. So we use the DISA flux and knockout cell, and we found that after we knock out the DISA, and the protective effect of notch was partially blocked. So that means notch pair law for DISA pair law for notch-mediated cardioprotection. So next, we want to know what is the key microRNA regulator by notch signaling. With the microRNA array, we found that MER322, 543, and 351 cluster is on the top list. Then we used this 3 microRNA cluster conditional mice, but it has like Z reporter. We found that this 3 microRNA is highly expressed in the neonatal heart, but it keeps at a modest level in the adult heart. So that is very important for the early heart development. And we found that this microRNA 322 express is also RPBJ dependent. When we knock out RPBJ, and the express was significantly reduced. And to know what is the binding site of notch, and we do the ChIP-seq, and we find that it is binding at about 6 kb front of this microRNA cluster. And this is a longer non-coding RNA. They express this 3 microRNA cluster. So this is the sequence notch binding. Then we do the overexpression experiment, use the lentivirus to overexpress MER322 and compare with the GFP. And we found that use the gene oncology enrichment, we found that it most regulate the angiogenesis signal pathway. And we also overexpress MER543 in the cell. We found use the RNA-seq. We found that it is most important for ventricular cardiac muscle development, also for the angiogenesis. So it is also help MER322 for improved angiogenesis. And finally, we want to know whole cluster, they are low in notch-mediated signal pathway. So we compared the GFP cell, notch overexpressed cell, and we used the local nuclear cell, local nuclear acid 3 microRNA inhibitor, and then do the RNA-seq. In this study, we found that this 3 microRNA play an important role for the arteriogenesis and skeletal muscle development, and also play some role for the epigenetic modification. And then we do want to know what is this MER322, their target, and we found that it has a Q2 as a target, and Q2 is E3 ubiquitinase intermediate HIF1 alpha degradation, and MER322 they can inhibit Q2. When they inhibit Q2, it can extend the half-life of HIF1 alpha. It is, we found that overexpressed 322, the Q2 level was decreased. And we verified the target by the luciferase activity assay. This is the Y-type 3' UTR. We can see 322 can inhibit the expressor of Q2. And when we use the anti-MER322, it can increase the expression, but it can be blocked by use Q2 mutation. And this experiment, we showed that when we overexpress 322 in the normal cell condition, it can upregulate HIF1 alpha level. But when we use the anti-MER322, it can further reduce the HIF1 alpha at normal cell condition. So in summary, notch activation can promote mesenchymal stem cell or the fibroblast cell, cardiac fibroblast cell differentiated into vascular smooth muscle cell in RPBJ-dependent fashion. And notch1 can promote cardiac fibroblast or cardiac mesenchymal cell mediate paracrine factor and improve angiogenesis and cardiac metabolism in ischemic heart. And we identified a novel notch1 downstream microRNA cluster, and it is important for the epigenetic regulation and muscle differentiation and angiogenesis. And I want to thank my research group and our Medical College of Georgia faculty that support, and this study is supported by 2 NIH HMBI grant and 1 muscle NIH grant and American Heart Association grant. Thank you for attention.
Thank you, Dr. Tang. That was a great talk. A quick question and quick answer. All right. If there are questions.
Nice talk. I just wondered, do these CMSC differentiate into endothelial cell or?
Yeah, they can increase CD31 DL4 expression. But most significant one is vascular smooth muscle cell, and smooth muscle cell heavy chain 11.
Oh, so eventually they differentiate into smooth muscle cells?
Yeah.
Okay, thank you.
Thank you.